Hello friends, welcome. So today we will be starting with microbes in sewage treatment. So we are talking about the chapter microbes in human welfare. We've already discussed how microbes were beneficial in the household, and also we have discussed how microbes were beneficial in the industrial areas. So next we'll go ahead and discuss microbes in sewage treatment. So coming to the microbes in sewage treatment, see basically sewage is something which is always there is a running out of water or municipality waste which is moving into the drainage. So this is a sewage which is basically municipal wastewater which contains a large amount of organic matter and microbes. Sewage is treated in sewage treatment plants, which is STPs, to make less polluting. So this water should not go out and it should not cause a harm to the society. Hence, what we do is the environment to keep it maintained, we treat the water before it is being expelled out into the river or the sea or anything like that. So it has got two stages. One is the primary treatment, another is the secondary treatment, which is the biological treatment. Now, coming ahead with the primary treatment. In the primary treatment, what happens? Microbes in sewage treatment, here in this case, in the primary treatment, the physical particles, the physical removal of particles will take place. So it can be in sequential filtration, which takes place one after another to remove out the particles which is there, or the floor, big say plastics or clothes or all those things whichever is there, they are being removed by using a filter, by using a mesh or something like that. And removal of grit, which is soil or small pebbles, very tiny ones, by sedimentation. They are allowed to sediment. Okay, they're allowed to the water is allowed to sediment so that the small particles will move down. The settled solids form primary sludge, and the supernatant forms the primary effluent. So because it is from the primary treatment, they are known as primary sludge, which is being removed, and primary effluent, which has been taken to the secondary treatment plant. So they are passed into a large aeration tank which is constantly agitated. You see, there's a constant mixing that is taking place. So the air moves in and there is a complete mixing which happens. This allows vigorous growth of useful aerobic microbes into flocks, which is bacteria associated with fungal filaments, to form mesh-like structure. So it is happening there, and these microbes consume the organic matter in the effluent. This reduces the BOD. What is BOD? BOD is biological or biochemical oxygen demand of the effluent. So the life and the chemical, whatever it's there, so the oxygen demand is being reduced. So this is known as, so there are flux which forms. So whenever there is an aeration agitation that is happening, you see there are flux that is getting formed. So when the flux are formed, so from the primary effluent, it has been taken into an aeration tank. Once it moves to the aeration tank, then after that the final clarifier will take place and it will circulate. So this will happen where the flocks are being formed. So there are flocks, if you see, a mesh like masses which are formed. All right. So that's happening. Next, we have the BOD, which is the amount of oxygen which is consumed by the bacteria to oxidize all the organic matter in 1 liter of water. So it is a measure of organic matter present in the water, and the greater the BOD, the more it is polluting potential. So the water will be more polluting. Hence, the BOD has to get reduced. So next we have this effluent is now passed into a settling tank. So here they are allowed to settle where the bacterial flocks are sedimented, and these sediments is called activated sludge. So now the flocks will sediment, and that is known as activated sludge. And a small part of activated sludge is pumped back into the aeration tank to serve as inoculum. Inoculum is a sample of the bacteria which is there. So that is being taken up. Okay. So this is how it is done for the secondary or the biological treatment. Now to see a gist on that, we have the rest of the sludge whichever is there. So the inoculum is taken, circulated into the aeration tank, and the rest of the sludge which is there, the remaining sludge is pumped into a larger tank containing anaerobic sludge digester. Here there are some anaerobic bacteria which digest the bacteria and fungi in the sludge by producing gases like methane, hydrogen sulfide, carbon dioxide. So all these are going to produce biogas. So now this activated sludge is used to produce biogas. This effluent is released into the natural water bodies like rivers and streams. Now the water whichever is there after the sludge is being removed is now free to move into the rivers or the sea, whichever water bodies. Now to put all the steps in a small gist, we see that first we have a primary effluent that is taking place where they are filtered or being settled, sedimentation is taking place. Then they are moved to the aeration tank. Once in the aeration tank, what is happening? There's a constant agitation which is happening. There is aeration that's taking place, and this aerobic bacteria is going to form flocks. So these flocks are going to reduce the BOD. So they are going to reduce the BOD. Now from here what we will do, these are being taken in and they are again led to settle because the flocks need to settle down. Sediment. So once they sediment, what is happening? Once they sediment, you take up the effluent and you take it to the settling tank, and from here the flocks are ready to get settled. Once they settle, then the inoculum, a bit of a sample is pushed into the aeration tank, and what happened to the rest of the sludge? The rest of the sludge moves to the anaerobic sludge digester where there is a digestion of the sludge, and thus there is a production of the biogas. And from the effluent tank, the effluent is moved to the natural water body. So that is how it is happening. So we see a pre-treatment, there is overflow that is happening, then a thrash removal that is taking place because of settlement, then air is passed and aeration is taking place, and then after that they come into the primary settlement tank where the effluent is taken and the sludge is again passed into the sludge digester, and the effluent is taken, aeration takes place, and after aeration they are allowed to settle. So then the inoculum is made to settle. A part of the inoculum is moved to the aeration tank and the rest of them to the activated sludge, which is moved to the dryer. Dry sludge finishing is taking place and we are getting a production of biogas. Now, here the Ministry of Environment and Forest has initiated Ganga Action Plan, Yamuna Action Plan to save water pollution. So there are various action plans which helps in how much impurity is there in the water, what is the oxygen demand which is taking place in the water that is there, and all those things. Next, coming to microbes in the production of biogas. So in the production of biogas, what is biogas? Biogas is a mixture of gases, mainly methane, which is produced by the microbes such as methanogens. Methanogens, they grow anaerobically on cellulosic material and produce methane along with carbon dioxide and water, which is methanobacterium. So there are a lot of methanogens which is there. Methanobacterium is found in the anaerobic sludge and rumen of the cattle, the stomach of the cattle, for cellulosic digestion. Biogas is used for cooking and lighting. Cattle dung, that is gobar, it contains this bacteria, so the dung is used to produce biogas. Usually all this organic waste. They are mixed with the dung of a cow and then after that allowed to settle for a few days to produce the biogas. Now, if I talk about a biogas plant, how does it look like? So there is a concrete tank which is around 10 to 15 feet deep. To collect the biogas and slurry of the dung, a floating cover is placed over the slurry which keeps on raising as the biogas is produced. So now once it is going to produce, there is an outlet that you see to supply the biogas. Now the outlet to remove the spent is also there, the slurry whichever is there. So that is being removed and that is being used as the fertilizers, which is biofertilizer. Now, Indian Agricultural Research Institute, IARI, and Khadi and Village Industries Commission, KVIC, developed technology of biogas production in India. Next, coming to microbes in bio as biocontrol agents. So, now in biocontrol means what? Biocontrol means is the use of biological methods for controlling plant diseases and pests. Example, ladybird, which you have beetle, which controls aphids. Dragonflies controls mosquitoes. There are chemical pesticides and fertilizers which kill both useful and harmful organisms and cause pollution. And biocontrol methods has no such problem. So they will selectively select the predators and will prey on the predator. Okay, next. Now to control butterfly caterpillars. So there are caterpillars. We have Bacillus thuringiensis. So now there are caterpillars which grows on the cotton plants. So to control it, the dried spores of BT, that is Bacillus thuringiensis, which is available in sachets mixed with water and sprayed on to the vulnerable plants such as brachias and fruit trees and all those. These are eaten by the caterpillar. In their gut, the toxin is released and the larva gets killed. So now, when the caterpillar eats this, the larva will get killed. Now, the scientists have introduced Bacillus thuringiensis toxin genes into the plants. Example, BT cotton, so that the cotton is not being infected by the caterpillar. Now, next we have Trichoderma species, that is fungus. They are seen in the root ecosystem. They control several plant pathogens that is there. Then we have baculoviruses, especially genus nucleopolyhedrovirus that we have, which attack insects and other arthropods. Now, it is very suitable for species-specific narrow-spectrum insecticidal application and desirable in IPM, which is Integrated Pest Management Program, to conserve beneficial insects. So they are very, very, species-specific. Next, we have microbes as biofertilizers. We were talking about the slurry. It's being dried and used as biofertilizers. Here we just have something more on biofertilizers. Biofertilizers are organisms that enrich the nutrient quality of the soil. So example, if we have, root nodules of legume plants, they will contain rhizobium, azospirillum. So they also will have help in nitrogen fixation. So and that is there. The free living bacteria, azospirillum, azoactor, they enrich the nitrogen content of the soil. Now, basically coming into mycorrhiza. Mycorrhiza are symbiotic association of fungi with the plants. The fungus gets food from the plants, and what the fungus does is to fix nitrogen for the plant. The fungal symbiotic performs the following: absorbs phosphorus from the soil, passes it to the plants, gives resistance to the root-borne pathogen and tolerance to salinity and drought, and it gives overall increase in the plant growth and development. And at the last, we have another bacteria, cyanobacteria, which is blue green algae. They are autotrophic microbes. They fix atmospheric nitrogen. Example, Anabina, nostoc, oscillatoria, etc. In paddy fields, cyanobacteria serves as biofertilizers. It also adds organic matter to the soil and increases the soil fertility. So that's it. Thank you. And just to brief up, we have seen all the places that microbes can be useful to us, whether it is household, whether it is industry, whether it is in the sewage treatment plant, then whether it is a biofertilizer, then there are various ways for nitrogen fixation in the biofertilizers that we have. So uh, there are various places where microbes can be useful as well. So thank you.